Greetings and welcome to the 2025 National Invasive Species Awareness Week. Today is our kickoff event, but this year's theme is focused on enhancing North American biosecurity to protect against invasive species. My name is Christy Trifone-Millhouse, the Executive Director here at the North American Invasive Species Management Association, also known as NISAW. Before we get started, a few housekeeping notes, as I was mentioning. Excited to see all of you today. Share with us in the chat where you're joining from. Also, after our presentations today, please feel free to use the reaction button to let our presenters know that you enjoyed today's topic. We'll be hosting a brief Q&A after the webinar. Questions do need to be added to the Q&A box. If you put them in the chat, we won't see them. All right, again, welcome. It's kickoff day for NISAW. The NISAW is an international event powered by NISAW to raise awareness about invasive species, the threat they pose, and what can be done to prevent their spread. NISAW provides a platform for individuals from local, state, federal, tribal agencies, regional organizations, private industry, and citizens to discuss legislation, policies, resources, and improvements that can be made with elected officials and decision makers. This is a week not only for learning, but for action. Throughout the week, we'll be sharing links with you in an effort to connect you with your U.S. senators and House representative, and we want them to hear our collective voices and to learn about the importance of our work and why we need support and funding to continue to press forward. For those who want to take a more active role this week, please head to NISAW.org and find the NISAW toolkit filled with template social media posts, press releases, PSAs, and more. If you aren't currently connected to us on social media, please join us and share our daily posts. As you can see, we have quite a week planned for NISAW with presentations focusing on our theme of enhancing biosecurity at all levels. Our first presentation today was from Dr. Dia Lawrence, and she really laid the foundation for our theme this week and talked about primary and secondary introduction pathway management. I encourage you to visit our website to register for webinars coming up. And if you weren't able to attend a webinar, we will be posting these presentations on our YouTube channel after the event. I would be remiss if I didn't mention that our agenda has changed quite a bit over the last month and continues to change. I would like to take a moment to thank those federal employees who have invested their energy and heart into this work. Some are no longer leading those efforts at the national level, while others are still holding strong. NISAW is driven by education and action, and as a partner, we recognize that now is not the time for all of us to take a step back and sit down, but rather as partners to stand up and move forward. I encourage you to activate your networks behind our calls to action and to provide support to programs and people that need a bit of glue right now in any way that you can. Through partnerships and collaboration, we are stronger. And this is the essence of our work here at NISAW. As leaders of NISAW is deeply vested in our mission to empower the management of invasive species across North America. We are led by an 18-member board of leading experts and professionals with supporting committees that I encourage all of you to join, a strong NISAW team, and a membership that continues to grow in numbers by the day. NISAW continues to be one of the most far-reaching events, with over 2.9 million people participating in last year's event. And we would like to reach even more in 2025. Here at NISAW, we provide the connections, the tools, and the voice that you need to address invasive species. We frame our programs around these three core pillars. We work hard to provide opportunities for collaboration and connection through our webinars, summits, our annual conference, and other events. We equip our membership and those in the invasive species community with the tools needed to manage and educate the public about invasive species. And finally, we strive to provide a collective voice on a national platform to advocate for relevance and support through our government relations programming and events like NISAW. Some of our upcoming events you won't want to miss include the 2025 National Forum on Biological Control in Annapolis, Maryland from April 14th through the 17th. The meeting themes revolve around biocontrol research, management, and impacts in natural areas. And we've got some great speakers lined up, so I encourage you to register today if you're interested in networking in person with members of the biocontrol community. Another event you won't want to miss is the 2025 NISMA Annual Conference. Mark your calendars and plan to join us in Lake Tahoe from November 3rd through the 6th. Our theme this year is charting a new course, and we've got another great lineup of speakers and really hope to see you all there. Head to NISMA.org today to register. As a friendly reminder, your membership dues help to make our mission come to life. And if you're not currently an NISMA member, we offer a variety of different memberships for everyone. You can view our new membership structure on our website at nisma.org. Want to sponsor NISMA in a bigger way? Our new Premier Partner Program is designed for you. Our Premier Partners enjoy benefits throughout the year, including acknowledgement on our website, at our events, and many other benefits. And I'd like to acknowledge and thank all of our current 2025 NISMA Premier Partners who generously made NISAW possible. Okay, it's now time for the main event, and I would like to introduce NISMA's Education and Prevention Manager, Tegan Wilmot, who will be moderating our webinar today. Tegan?
Thank you, Christy, and welcome to everybody. As Christy said, I am Tegan Wilmot, the Education and Prevention Manager at NISMA. And I would like to first start by introducing our speaker here. We have Joe Krieger, and he is NOAA's National Invasive Species Coordinator. He will be presenting later on NOAA's response to an emerging threat of invasive soft corals in the Pacific and Caribbean basins. So Joe, if you would like to give a brief overview of NOAA, I'll turn it over to you now.
Well, good afternoon, good morning, good evening, everybody. Thanks for tuning in to this webinar. I'm excited to be here and to talk a little bit about NOAA's work to help support biosecurity, both in the marine setting and also in the Great Lakes environment. My name is Joe Krieger. I'm NOAA's Invasive Species Coordinator. I am located at our Great Lakes Environmental Research Lab in Ann Arbor, Michigan. And I just wanted to start things off by just giving kind of a brief overview of NOAA for those that aren't familiar, just kind of set the stage for the rest of this afternoon's presentation. All right, so NOAA, or the National Oceanic and Atmospheric Administration, is part of the U.S. Department of Commerce, and our mission focuses on science, service, and stewardship. Specifically, our mission is to understand and predict changes in climate, weather, oceans, and coasts, to share that knowledge and information with others, and then to conserve and manage coastal and marine ecosystems and resources. And so why is NOAA concerned with invasive species? Just to put it shortly, it's because we are directed by executive orders and congressionally mandated to participate on a number of national and international collaboratives. We hold key leadership roles in two national level invasive species organizations, which many on this call are probably familiar with, both the National Invasive Species Council and the Aquatic Nuisance Species Task Force. NOAA also has leadership roles on a variety of national and international strategic efforts. The most relevant to this particular topic would be the participation in the Regional Biosecurity Plan for Micronesia and Hawaii. And so just a brief overview of the National Invasive Species Council for those that aren't familiar. This was a group that was established out of a couple of executive orders. It's a White House level group. NOAA serves on behalf of the Department of Commerce as one of the co-chair agencies, along with the Department of Interior and the U.S. Department of Agriculture. This group is comprised of senior leadership from 12 different federal agencies and 4 executive offices of the president. And really, its goal and mandate is to manage invasive species at the national level and provide policy and guidance for this group. It coordinates its priorities through an annual work plan, which is updated annually to address new priorities and issues that arise. And the other group I mentioned was, and you might be already familiar with this, is the Aquatic Nuisance Species Task Force. This group was created out of an act of Congress under the Non-Indigenous Aquatic Nuisance Prevention and Control Act. NOAA co-chairs this along with the Fish and Wildlife Service. There's 13 federal agencies, 15 ex officio members. And really, this is more of the boots on the ground folks that are actually out in the water, the weeds, enacting these invasive species management. They're also made up of a number of regional panels that bring into the states, the tribes, the territories, and the private sectors. Yeah, so those are the 2 main groups that NOAA helps to coordinate. And really looking at how invasive species impact NOAA and its mission. What is the nexus of invasive species and NOAA? So invasive species do have profound impacts on some of NOAA's mission and managed resources, looking at things like protected and endangered species, impacts to our national marine sanctuaries, which you're going to hear about a few examples of that a little bit later this afternoon, impact to aquaculture operations, essential fish habitat, coastal fisheries, wildfires, fire weather predictions. All of these things have some sort of management obligation to NOAA, and invasive species are impacting those. They also impact NOAA's ability to promote this idea of the blue economy, which is a big part of NOAA's mission. And then thinking about just where these impacts are happening, this is definitely at a national level for NOAA. It affects all of our line offices at varying degrees. We're going to talk a little bit at the end of this webinar about this invasive soft coral issue that's happening both in the Caribbean and the Pacific Basin, which is really bringing in a lot of NOAA expertise. We've identified all of our line offices, like I said, have some sort of crux with invasives, whether it be invasive grasses or wildfire, marine invasives, coastal invasives. NOAA is really engaged in these efforts. Some of these broader national initiatives, such as the Early Detection Rapid Response Framework, NOAA is also very instrumental in parts of that. And then we also more recently have joined and formed collaboratives with the U.S. Coral Reef Task Force, which is the federal national response management authority for our nation's coral reefs. So just kind of wanted to give that high level perspective about why NOAA is involved and where it is impacting our mission. And then we're going to go into some more specifics later this afternoon on some of the work that we're doing towards biosecurity. And so I just wanted to briefly just introduce our presenters. They'll probably get introduced in front of their talks, but I'll just go ahead and do it right here. So next up, we're going to hear from Valerie Brown, who's part of our Marine National Sanctuaries system. Then we'll hear from Rochelle Sturtivant, who's with Michigan Sea Grant and NOAA Great Lakes Broad Research Lab. Michael Studivan, who's with NOAA Oceanic Atmospherics Research. Then we'll hear from Brian Hauk, also with National Marine Sanctuaries. And finally, Steve Gittings, who is also with National Marine Sanctuaries. So with that, I'll stop. I'll turn this over to Val, and we can get going on the biosecurity focus portion of this presentation. So thank you.
Thanks, Joe, and talofa lava, everyone. As Joe mentioned, my name is Val Brown. I'm the research coordinator at the National Marine Sanctuary for American Samoa, and also the co-lead for the Pacific Preparedness Team of the U.S. Coral Reef Task Force's Coral Disease and Disturbance Working Group. Happy to be here with you today to talk a little bit more about our efforts to improve biosecurity after facing some recent some challenges out here. So in my role as the research coordinator for the National Marine Sanctuary of American Samoa, I'm tasked with implementing the conservation science and resource protection programs for one of the largest sanctuaries in the U.S. I support research and monitoring, as well as evaluating risks to our sites from a range of threats. Part of that portfolio includes safeguarding the spectacular places like the Valley of the Giants and Fungatelli Bay from invasive and nuisance species. In 2019, though, we learned that a disease referred to as stony coral tissue loss disease was spreading from Florida across the Caribbean, and it caused dramatic declines in coral populations. SCTLD is particularly frightening prospect for the islands because it attacks corals, the very foundation of the reefs surrounding the islands, and Pacific reefs have tremendous biodiversity and value. These corals provide habitat for over 1,000 reef fish species and thousands of invertebrates, which feed island communities and support tourism. The reefs also buffer storm waves. They reduce about 97% of the total wave energy approaching our shorelines, and the corals constantly grow to keep building and cementing the reefs together. They're living coastal defenses, natural infrastructure that protects our coastlines. And this is a valuable service for places like American Samoa because most of our villages lie close to the water and a very thin strip of flat land between the shoreline and soaring mountains. You can see here how the reefs break up the energy far offshore and protect these vulnerable areas. Reefs in the Pacific also protect tourism, port infrastructure, and military installations. The scope and the impacts and the rapid spread of stony coral tissue loss disease across the Caribbean triggered alarms out here in the Pacific and resulted in the creation of the Pacific Preparedness Team within the broader U.S. Coral Reef Task Force's Coral Disease Working Group. We started by building capacity and disseminating scientific information from affected jurisdictions to the Pacific and developing initial Pacific surveillance guidelines. We also shared disease sampling and intervention guidance, conducted trainings, and the goal was to raise awareness of this potential invader and prepare teams with the best scientific guidance and tools to act in case it was ever observed in the Pacific. This disease moves really quickly, though, and so far, intervention efforts have proven costly and time-consuming. Once that disease takes hold, it's hard to stop. And by late 2022, there was mounting evidence to support the hypothesis that the disease was moving via both short-range pathways, such as direct contact, water movement along individual reefs, as well as longer range pathways, likely moving with vessels. And that can be through one of two different mechanisms, possibly others, but the first one is hull fouling or biofouling, which is the accumulation of marine growth on ship hulls. Although vessel operators try to keep ship hulls clean for better fuel efficiency, there are recessed areas on vessels that seem to attract fouling organisms. This is particularly true for vessels that sit for a long time. There are no international standards for this. There is guidance, but it's pretty poorly studied, and there's because there's no actual agreements, there's no enforcement of these except at local jurisdictional levels. However, there have been numerous documented cases of species introductions directly linked to hull fouling, which makes it concern. The other perhaps more familiar vector is ballast water. So ballast water is carried by ships to offset their loads to maintain vessel stability. When the ship unloads cargo, they fill their tanks with water to maintain their weight for safe operation. They then travel to their next port, and as they load cargo, they discharge that ballast water as they take on more weight. The ballast water is a known vector of invasive species, including pathogens like bacteria and viruses, as well as larger organisms. The risk is not just the water itself, but could also be biofilms within the ballast tanks or sediment that's retained in the tanks. There are guidelines to help manage these processes, including an international convention to minimize risk, but there is little testing of the ballast water to ensure that those methodologies are meeting the needs, and there's limited enforcement as well. And so research by NOAA and others suggests that the other issue is that research by NOAA and others suggests that some of the approved technologies, particularly UV treatment systems, may not be sufficient to address pathogens like stony coral tissue loss disease. And so when the science confirmed that these pathogens could be moving via ship, we realized maybe we should be thinking about this in terms of an invasive species, or more specifically, an invasive pathogen. So it started looking bigger. And as the Coast Guard started evaluating vessel movement of ballasted ships across the Pacific, they alerted us to the fact that vessels were traveling through the Panama Canal to Hilo, Hawaii, and American Samoa after stops in affected jurisdictions. And this really brought us to this concept of marine biosecurity and thinking broader about this as an invasive species, how we might address it, and what science we needed to improve that marine biosecurity effort. And you can see here, all of this bright green is vessel tracks in the map. So you can see just how connected the entire region is by vessel traffic. In fact, the entire world. I do want to make it clear that stony coral tissue loss disease has not been detected in the Indo-Pacific at this time, and the likelihood is unknown. The preparedness efforts, though, have highlighted gaps in marine biosecurity across the region. For those of you that aren't familiar with the term biosecurity, you can think about it. I've put up a couple of definitions from plans in the Pacific region, but you can really think about biosecurity as an approach or a system that focuses more on the mechanisms of introduction with a focus on preventing or then detecting and minimizing impacts. It considers the risk to ecosystem functions as well as the ecosystem services and impacts to people and economies as well. There are a number of talks this week about terrestrial biosecurity, but unfortunately, marine biosecurity efforts have lagged behind. They're challenging to implement, and the marine environment is difficult to implement containment. One of the efforts that has looked at marine biosecurity was the Regional Biosecurity Plan for Micronesia and Hawaii. This document was released in 2014, and it was the first regional assessment of marine biosecurity in the Pacific. It was associated with military developments in Guam, so it analyzed the connections between Guam, Commonwealth of the Northern Mariana Islands, all the way to Hawaii, and then all of the ports that connected to those jurisdictions. The assessment found a nearly complete lack of capacity for marine biosecurity throughout the region and confirmed that invasive species are a significant threat to regional economies that depend on fisheries and tourism. It also recommended that efforts to improve marine biosecurity should focus on prevention, detection, and rapid response. Many of you may be familiar with this graphic. So this is the invasion curve. It shows the progression of an invasive species over time, crossed with cost and area infested. And what has held true time and time again is that the best option from both an ecological and an economic perspective is prevention, to prevent the spread of an invasive species before it gets to new places. And if that fails, it's important to have early detection and rapid response to quickly eradicate and control the invader. This requires research and development of surveillance and monitoring tools, as well as response and control methods. These early responses are particularly important because in the marine realm, containment can be incredibly difficult to achieve and may not be possible due to water currents and movement and just the connectivity of the marine environment itself. When we think about stony coral tissue loss disease, the Pacific is still in the bottom left-hand corner. So we can still focus on prevention and early detection and rapid response. But in other locations, the disease has now shifted into long-term management as it becomes endemic. Oops. So One of the efforts that has been underway recently has been to kind of assess the research needs for marine biosecurity. And so this list here was a prioritized list from an invasive species forum held in Guam in 2023 and hosted by the Department of Defense. With this, they noted a few key priorities for science needs that folks are now trying to work to address. For prevention, that included evaluation of ballast water management systems to see whether they're actually effective against pathogens, improving techniques for the management of biofouling in those niche areas we looked at earlier. It also focused on early detection and rapid response. So utilizing new tools like AI and machine learning to detect new marine invasives quickly and effectively, to develop genetic and molecular database for marine species of concern, develop new tools for response and control of invasive species if they do invade, and to improve available strategies and tactics for response. And I do want to note this is just an example to kind of show why biosecurity is important with an example from one invasive species. There are many invasive species out there that NOAA is addressing. There are also other modes for introduction, including aquarium releases, aquaculture, and transport by smaller vessels. You'll see in the following talks some examples of how NOAA scientists are working to address invasive species and marine biosecurity across this entire spectrum of the invasion curve. We'll hear about new information systems, horizon scanning, molecular tools, citizen science, best management practices, and improved partnerships to safeguard America's oceans, and the social and economic benefits that they provide to our country. And with that, I'm going to wrap up and hand it over to our next speaker, Rochelle, so you can learn more about all of NOAA's efforts. Thanks.
So, start off with, I'm Rochelle Sturtivant. I am the program manager for NOAA's Great Lakes Aquatic Non-Indigenous Species Information System. So I do work out of the Great Lakes rather than in the marine environment, but the systems are parallel with many of the same invasive species issues. I am a NOAA affiliate, not a federal employee, and I'm with Michigan Sea Grant, which is a NOAA affiliate program, and I'm an employee of Michigan State University Extension. And my clicker is way over on the left side, so I apologize if I'm turning away from everybody. Okay, and it's not, there we go. Wanting to start off with a huge thank you to the National Invasive Species Council, who has a task team that is working on a paper on horizon scanning that it has been my pleasure to work with for the last year, slightly over a year now, on a paper on horizon scanning. So I'm going to start off with some definitions that were provided by the National Invasive Species Council. We've really worked through hammering out distinctions between what we call horizon scanning and what we call risk assessment, which chain together, and many people, the line blurs between the two sometimes. But horizon scanning is defined as the assessment and qualification and or quantification of the risk of invasion by non-native species into a defined geographic area through analyses of available data. The end result of a horizon scan is an extensive list of potential threats. Risk assessment, on the other hand, tends to look at one species at a time. It's a systematic, extensive, and thorough review of the species' potential risks to a specific jurisdiction. It identifies as its end product the relative risk posed by that particular species to that particular situation. I'm going to talk about both horizon scanning and risk assessment in context of our work in the Great Lakes. So horizon scanning, using this figure that Val introduced to everyone, horizon scanning takes place way on the left side of this invasive species introduction curve. This is the very first step of figuring out what is it we're trying to prevent. So it's a pre-prevention step. which pathways and species are posing a threat to your jurisdiction? In our case, a threat to the Great Lakes. And then the horizon scanning effort is followed by risk assessment, digging into more of the details specific to our region. So I'm working, sitting at NOAA's Great Lakes Aquatic Non-Indigenous Species Information System. And one part of our website is a clearinghouse of risk assessment information for the Great Lakes. This was developed in around 2021 at the request of regional management agencies through the Great Lakes Panel on Aquatic Nuisance Species, which is the regional panel under the Aquatic Nuisance Species Task Force for our region. We basically went out to the management agencies and said, hey, what are you guys using for risk assessment? You know, when somebody comes to you with a permitting decision, or that you're trying to figure out, you know, what's a threat that you should be watching for, setting up your early detection programs. That generated for us a list of 16 assessment and horizon scanning methods that they were already using. These methods range from global scans like STARE, based just on the species biology, how likely is species X to move, basically, is it spreading globally, those types of questions, down to some really state agency specific assessments, such as those conducted by Wisconsin DNR, that are focused very specifically on the issues for that particular state, and for, in particular, their agency trust resources. Others are focused in this group on taxonomic groups. For example, there is an assessment of fish or an assessment of crayfish that might be a threat. Yet others are pathway. There's one of the assessment horizon scanning exercises was done for ballast water for the Great Lakes. So we put together this set of 16 and developed this clearinghouse so that everyone who's working in the Great Lakes region can see all of the information that each other are focused on and using in their day-to-day work. When we had it together, we realized that we had a wonderful resource to do a horizon scan for the Great Lakes. This was a huge pile of work done by a whole lot of other people. And we wanted to just take a look and see how many total species is that, and are they they're threats to the region. So these are the sources that we used for that. Again, they include everything from global to national, right down to very state and provincial specific methods. There's 16 separate applications of these methods. So some of the methods were used in a couple of different applications. Altogether, that has a total of over almost 5,000 species assessments. And then some methods did the same species. So it's actually just over 3,000 unique species that are in this database that someone has identified as proposing some level of risk. That was the starting point for our horizon scan. And we moved from that into a rapid screening method. And again, this is very specific to the Great Lakes and very specific to the particular database that I manage, the Great Lakes Aquatic Non-Indigenous Species Information System. So the first thing we did with our rapid screening is it one we already have, that we already know about, we're already working on, and we took those out. And we were already actually looking at 294 of those species. And then we applied our criteria. So Glancis is aquatic. There were a lot of terrestrial species in here. Glancis also does not include birds and mammals because we consider them to not be normally living submerged in the water. And so that took out a chunk. We also found that there were several that had been, particularly, I think they came through the global horizon, the global scans, that were already native to the Great Lakes. They might be a high risk somewhere else, but they're our native species. So we took those out. And again, this is a rapid screening process, trickling these down step by step to a manageable number that we could consider actual threats to the Great Lakes. Some of these, the original assessments that were included actually scored them as low risk. So we took those out. And then finally, the biggest chunk that we were able to remove from this list were climate mismatch. Again, these were mostly coming from those global level scans, and included a lot of tropical species that are not going to be able to overwinter in the Great Lakes. So we took those out. That still left us with a horizon scanning list that's a feasible threat to the Great Lakes region of almost 800 species, which was a lot. It was far more than we were expecting we would end up through this screening process. From that point, the plan was that we were going to just take a random subset and run through our risk assessment and see, whether once we ran all the way through our risk assessments, those would be still be considered a threat, a high risk to the Great Lakes region. We weren't quite random. In fact, we pulled out 3 specific subsets. The first was reptiles and amphibians. Those had not been previously included in our database. We got very clear direction from our state agency partners that they would really like us to include those. So we put those to the top of the list. They also requested some pretty extensive sets of species that they specifically wanted us to look at. And so those came to the list. And then a partner of ours, Alicia Davidson, who works with she's an independent consultant who works closely with the Nature Conservancy and was currently doing a project for the Great Lakes Panel on Aquatic Nuisance Species. Provided us with copies of assessments that she had done for a subset of this species that were done on a template that was like only 2 questions different from our core risk assessment process. And so we prioritized those as well. So we wound up looking at 128 species of this 795, and we set the other 667 aside for now. And this was the group that then we moved into our risk assessment process. So we took that set of species, we looked again, tried to look at whether they were native. 2 species fell out as cryptogenic. We aren't really sure, even going through all the literature we could find, they may or may not be native to the Great Lakes region. Then we looked at, there were a small set of species that had a history of failed introductions, meaning they've been introduced to the Great Lakes. We have records of them being introduced, and yet they've never become established. They've never been able to overwinter or reproduce. So those were fairly quickly eliminated. And then we started running through our very detailed risk assessment, looking at the probability of introduction. And that looks at whether there is a pathway from a source population into the Great Lakes that has some probability of carrying that species from point A to point B, from somewhere outside the basin to somewhere inside the basin. That actually only eliminated 7 of those 128 species as having low probability of introduction. And then we really started digging in on the biology of these species and determining whether or not, could they find food here? Could they find habitat? Is the temperature tolerance within their range? And that's where we really ended up kicking out a lot of the species in this set that had been through the horizon scan were a risk. But when we really dug in on the biology of the species, we eliminated a lot of them because some critical factor was missing that species required. And so that got us down to a list of 15 species that we felt were a credible threat to the Great Lakes and we would add to our database. And these are that set of species. I just wanted to throw those out there for you real quickly. Two of them ended up actually having already become established during this process, or in fact, in the case of Salix cinerea, well before. They've probably been here for 100 years. We just hadn't realized they weren't native. And the Anguillicola crassus is a nematode parasite of American eels. So those two are actually already here. There were two that we put into kind of a range expansion cryptogenic category. That includes the red-eared slider and the American bullfrog. They're probably native to the southern edge of the Great Lakes Basin, but the Canadian populations and the northern populations are very definitely not native in those areas, and these species have been exploding. And then the rest of these here were added to our watch list. So that goes then into the hands of the folks in this region who are out in the field doing those early detection efforts to hopefully catch these species and identify whether or not, in fact, they are being seen, whether they're being introduced. And if so, to be able to find them and get on top of those populations quickly to prevent establishment. I do want to point out that our Great Lakes effort, while quite extensive, does have a national counterpart through the Early Detection Rapid Response Framework, which is headed by primarily Department of Interior, but with buy-in from all of the federal agencies. It's a true interagency partnership that will include, when completed, all of the terrestrial species, all of the aquatic species, all of the marine species, all of the wetland species, etc. And just a quick look, Siren just a couple of weeks ago went live in beta for those who are interested in finding resources for your own regions. It combines them, as I said, the marine, Great Lakes, inland freshwater, terrestrial. I believe wetlands is included as well. Horizon scans. Ours is not in the database yet. The regions that are shown here as having horizon scans is reflecting right now just the horizon scans that were conducted, I believe, by Fish and Wildlife Service. But anyone who's done horizon scans, there's opportunities to add your data into this database. Species profiles and risk assessments for the entire nation, again, for all of these regions, for all taxa, are slowly coming online as different agencies have worked on different sets and are contributing to that. Our Great Lakes ones are not included yet, but I am in the process of working with them to figure out how we can link our resources into this national database. And they've also recently restarted an experts database under the Siren header. This previously existed under the General ANS Task Force website, and it's been handed off to Siren as part of this national framework. And it's building very rapidly, but will allow you to figure out who are the experts for particular species, for particular taxa, for particular regions. It includes folks who are experts in taxonomy, experts in the ecology, experts in control of species, folks who've done case studies on different control methods. All of those kind of experts are putting their contact information into this database. So it'll be a wonderful wealth of resources for people. And I believe that is all I have. I do want to acknowledge that obviously this is not all my personal work. GLANCIS is a NOAA product and funded by the Great Lakes Restoration Initiative. We're partnered with USGS's NASS program. NASS runs the National Inland Freshwater Database for aquatic invasive species. And the Great Lakes, our Great Lakes database is piggybacked on top of that. The Great Lakes Panel on Aquatic Nuisance Species both provided input and support for the development of that of our original horizon scanning and the risk clearinghouse. The Cooperative Institute for Great Lakes Research has provided us several summer fellows, as has the University of Minnesota Duluth. Myself, I'm a Michigan Sea Grant staffer, as well as 3 other members of the GLANCES team are Sea Grant staff members. Some of them through, most of them through the University of Michigan. The Great Lakes Commission provided funding as well for the development of that clearinghouse of risk, and several staff members from Sigler, Gloss, without whom this work would not have been possible. So thank you very much for your attention, and I will pass it to the next speaker.
Thank you, Rochelle. I'm just sharing my screen now. I'm excited to be here today. I'm going to be bringing us back into the marine realm, specifically focusing on stony coral tissue loss disease, as Val gave a great introduction to. My name is Michael Studivan. I am a researcher with the University of Miami, as well as NOAA's Atlantic Oceanographic and Meteorological Lab in Miami, Florida. So before I jump into the bulk of my talk, I wanted to first thank this amazing group of collaborators and team members that have worked on this project, spanning 6 different partner institutions, including AOML and University of Miami. None of the work that I'm presenting on today would have been possible without the amazing efforts of all of these folks. As Val mentioned, stony coral tissue loss disease is kind of a major event. It has, since it was first observed, it has now become the most devastating coral disease ever described. It affects over half of all the reef-building coral species in the Caribbean. It is up to 34 different species as of right now. It causes rapid mortality, where tissue loss just migrates across a coral colony. And generally, that colony dies within days to weeks. It has been completely unprecedented in terms of both the scale at which it's moving from reef to reef, but also across the wide variety of diverse coral taxa that it affects. It was originally observed off of Miami, so a couple miles from where I'm standing right now, in 2014. Over the last 11 years, it has now spread throughout the majority of the Caribbean. This map here is 2 years out of date, and unfortunately, instead of being 25 jurisdictions that the disease has been observed in, it has now been observed in 33 different jurisdictions, where it's starting to curl. I'm not sure if you can see my mouse, but it's starting to curl around in the southern Caribbean. And what is particularly interesting about this disease is its transmission across jurisdictions hasn't necessarily followed oceanographic patterns. As we're starting to see this southward march of the disease observations into the Caribbean, it was going countercurrent. And as Val mentioned, we suspected that the disease was largely moving through water via currents, but now we are starting to believe that the disease could also be moving through human-mediated transport, such as through ships' ballast water systems or on ship hulls as biofilms. And so it's become a huge problem for the entire Caribbean basin. But what I'm going to be focusing on next is not actually in the Caribbean per se. It's this tiny little spot right here. So if you don't recognize it, I'll take a little zoom in. This is the Panama Canal. It is a roughly 50-mile stretch of freshwater lakes, canals, and locks that connects the Atlantic Ocean from the Pacific Ocean. In 2023, nearly 15,000 vessels transited across this stretch, bringing cargo to and from the 2 different ocean basins. But they are also bringing ballast water from one ocean to another. And so this represents a significant threat to biosecurity. As Val mentioned, there have been numerous species introductions, but it also could be a potential route for disease pathogens to be transported. If we take a look at the shipping activity on any given day at the Panama Canal, we see a lot going on. There are ships that are constantly transiting in either direction through the canal. There are anchorage zones shown with the dots on either side. And I want to focus specifically on one of the anchorages on the Pacific side here. There's a series of islands as part of the Pearl Islands off of Panama. And if we take an even closer look at just one of those islands, we can see a lot of well-developed reef structure here on that northern coast. That is dominated by branching species such as Passalapra, shown here on the right, and it is inherently linked with commercial shipping operations in the area. Indeed, on the same island, there is a refueling station that a lot of these tanker and container ships dock at. So they are a stone's throw away from the nearby coral reefs. So circling back to here, I want to contextualize this in terms of the invasion curve that Val presented. In the Caribbean, we are very much at the long-term management response with respect to SCTLD. It's pretty much spread throughout the entirety of the Caribbean, but importantly, as she mentioned, it has not spread so far into the Pacific. And our mitigation measures have largely failed. And so why? Why has disease mitigation efforts failed in the Caribbean? And how can we learn from that to be better prepared to hopefully prevent SCTLD from spreading to the Pacific? So the short answer is that disease mitigation is really tough. There are over 40 different coral diseases that have been described to date, and of all of those, we know the pathogens of maybe 2 or 3. Corals, despite being an evolutionarily simple animal, they're extremely complicated when it comes to how they interact with their environment and how they interact with other microbial constituents. I'll get into that in a little bit, but what I want to focus on is how the AOML Coral Program here in Miami is addressing some of the failures in disease mitigation related to SCTLD so that we can better prepare other regions like the Pacific to be able to respond in case there is some type of disease transmission. So all of our work starts at the top with transmission experiments, because we can't just go out on a reef and expose corals to disease and see what happens. We need to be able to control every aspect of their environment so that we can specifically identify how a disease could be transferred from one coral to another, whether that uses some type of non-biological intermediary. But we also need to be able to better understand how our treatment approaches are working or not working in a lab-based setting. So first and foremost, we run a whole lot of transmission experiments so that we can better simulate what could be happening out on the reef. From there, we collect samples so that we can better understand how those corals are responding, particularly at a cellular level. Using cellular diagnostics, such as tissue histology, we can better understand what is happening to the coral, what is happening to their algal symbionts, so on and so forth. We can also use a variety of different molecular techniques by looking at the DNA and the RNA found within the corals, as well as their symbionts, to learn how they're responding to that disease exposure. So this generates a lot of data, and it tells us roughly who's there, who's not there. But from there, we need to translate that and distill it down into identifying specific taxa or specific genes that can be quickly and cheaply used to diagnose disease. So can we screen for SCTLD exposure, or on the other side, can we screen for disease resistance in individuals so that we can make more informed decisions? And then finally, bringing everything that we learn together from these various different focal areas, being able to use adaptive restoration techniques so that we are putting disease-resistant and stress-resilient genotypes back out onto the reef to promote the recovery of that reef population. So I'm going to walk through examples from each of these with a specific focus on some of the work that we're doing with Pacific coral reefs to see what we've learned so far. In order to be able to draw these really important conclusions from our work, we need to have a really good setup for exposing corals to disease in the lab, where it poses very little risk to spread beyond our lab, as well as allowing us to have a high degree of accuracy and precision with respect to how we're exposing these corals. So we developed this disease transmission apparatus at the University of Miami, where every single coral has its own independent water source. This is very important because it allows us to run experiments in a statistically robust and independent manner. This means that a nearest neighbor doesn't influence whether or not a coral gets diseased. So every time we see disease, we know it's specifically from what that coral is exposed to, from nothing else. So there are these half-liter coral vessels that each hold one coral. Each one has an individual water source, and all of these are maintained in a water bath that allows us to keep environmental parameters consistent. If we take a top-down look over on the right-hand side, we can see little coral fragments in each of these jars. Sometimes we also use sediments as a means to expose corals to disease. Sometimes we also use other coral fragments themselves. So recently we conducted an experiment in this setup using 3 different species. We had 2 Pacific species, Pavona clavis in the top left, Passalopra, a branching species, in the top middle, and then we compared that with a Caribbean control in the top right. That's Orbicella favulata. It's known to be moderately susceptible to the disease. When we expose these 3 species to stony coral tissue loss disease, we see rapid tissue loss. It happens almost overnight in some cases, and it manifests very similarly across the 3 species. So this doesn't look great off the top right there. And then we can also quantify the susceptibility of every single fragment to disease, depending on how long it took to develop disease signs. So I, of course, need to point out, none of our corals exposed to healthy water experience any kind of tissue loss. All of them survived. However, when we expose corals to disease using 2 different means, either waterborne disease or sticking a diseased coral next to it, the corals quickly start developing lesions and succumbing to that disease. So it's not looking too hopeful. We then use our cellular diagnostic methods like tissue histology to better characterize what is happening inside that coral tissue. So this panel here is showing healthy corals of Passalopra on the top, Pavona clavis on the bottom. We can see a couple things that are pretty typical in healthy corals. We see the epidermis, or their outer tissue layer, the mesoglia, that layer in the middle, and then finally the gastrodermis, which is that inner layer, which also hosts algal symbiont cells of the family Symbiodiniaceae. Now, if we compare that with some slides taken from disease or presumed disease corals, it's a very different story. And you don't need to be a histologist to say that something is going on here. These corals look pretty bad, where we have evidence of necrosis, or the tissue just falling apart. We have body wall breakage occurring. We have those symbiont cells getting exocytosed or ejected from their tissues. Separation of the gastrodermis from the epidermis and mesoglia, and then finally vacuolization. Normally, these algal symbiont cells are held in these little tissue compartments, and as vacuolization occurs, those compartments swell up larger and larger. And I want to dive a little bit deeper into this because I'm going to take us back in time a little bit and focus on some of the work that we've done in the Caribbean to better characterize what's happening in our Pacific experiment. So as I mentioned, a lot of these processes are happening, and they happen all the time in healthy corals. It's part of natural cellular turnover, similar to the way that our cells regenerate once they become old. We have vacuolization, where those algal compartments get bigger. Some symbionts might be degraded or digested inside the coral tissue. Others might be ejected. And then finally, we sometimes get separation of the gastrodermis from the mesoglia and epidermis. In disease corals, we see all of these happening, and they're all elevated. So every single one of these processes is kind of happening in a runaway fashion. And so together, it's called a process known as symbiont dysbiosis. And we think that's what is happening when corals are exposed to SCTLD. Something is happening where there's a breakdown of communication between the coral host and the symbionts. And to put it mildly, all hell breaks loose inside those coral tissues. By looking at all of these different metrics, we can start to quantify them in healthy versus diseased corals. And what we found is that vacuolization, or the size of those tissue compartments, actually corresponds really well to overall disease state. So as those vacuoles get larger and larger, eventually you get to a point where more than likely that coral is diseased. And where this really comes into play is being able to differentiate diseases like SCTLD that have this characteristic vacuolization from another disease like white plague. So white plague and SCTLD look remarkably similar in the field. If you were diving and you saw two coral colonies, one with one disease, the other with the other, you would not be able to tell the difference. But under a microscope, we can, and we can use these tools to differentiate them so that we can apply disease-specific treatments to be able to help save that coral. Switching gears a little bit into the molecular side of things. I'm going to keep it pretty high level because I know that this is pretty much the nitty-gritty, and it is a lot of the stuff even goes over my head. Now, as I mentioned before, one of the challenges in being able to identify what makes a sick coral sick is the fact that corals are extremely complicated. They host a diverse suite of microbial symbionts that includes the algal symbionts I talked about earlier, as well as bacteria, viruses, fungi, and other microeukaryotes. And we don't really have a good handle about what makes a normal symbiotic relationship for corals. And so we're applying a suite of different molecular tools, including metabarcoding, metatranscriptomics, and coral transcriptomics to identify what makes a healthy coral healthy. Once we do that, we can then compare that to diseased corals. So we can learn what specific bacteria or viruses are in different abundances. We can learn how the corals are responding differently to that, to be able to distinguish what makes each disease different from one another. And so to date, we've amassed over 500 samples throughout the Caribbean with a big focus on SCTLD, but in reality, we have 10 different coral diseases and 13 coral species. So as I mentioned, this includes SCTLD, also other white syndromes like white plague, white band, as well as other diseases, including black band disease, red band disease, dark spots disease, aspergillosis. To our knowledge, this is the biggest coral disease data set ever assembled from one specific area and will allow us to specifically identify what similarities and differences exist. Across all of these different diverse taxonomic relationships, the coral species that we've sampled, we have a series of different tissue samples. We have apparently healthy in advance of where that disease is occurring, the transition line at that lesion margin, and then finally dead skeleton to look at who's colonizing after the fact. And then we have healthy conspecific samples from nearby so that we can do those healthy disease comparisons. Okay, so from this, we've generated terabytes of sequencing data. We know thousands of different microbial partners who are there, who aren't there. How do we make any sense of that? So from here, we use a process to look for potential biomarkers. These are indicators of disease or indicators of resistance. As part of a collaborative project with Mote Marine Lab here in Florida, we conducted the largest coral disease transmission experiment to date, where we exposed hundreds of fragments of Orbicella faveolata to stony coral tissue loss disease. And we tracked these fragments over time to look for disease signs. We also collected samples all throughout, before disease, right after disease, at the sign of first lesions, and then finally when they were dying. And from there, we can look at the overall bacterial communities. So in this stacked bar plot off to the left, we have columns that correspond to that sampling time point, pre-exposure before disease, immediately after, the initial disease lesions, and then final. And the colors correspond to different bacterial groups. It's a lot to take in, I get it. But what we can do is focus on those specific bacterial taxa that increased in abundance as they were exposed to disease. And then we plot that with our different genotypes that were used in the experiment, and we can identify certain individual bacteria that seem to be driving a lot of that variation. In this experiment, we found this Rhodospiralis that explained a lot of the variants. So this could be a bacterial taxa that we could develop into a rapid assay so we could quickly screen individuals out in the field and determine whether they've been exposed to SCTLD or conversely, whether they are resistant to this disease. And then finally, to bring it all together, of how we can take what we are learning about this disease at the cellular level and translate that into management action. Well, those same Orbicella fravulata fragments that were used in that previous experiment I talked about are in active propagation and outplanting as part of Mote Marine Lab's restoration initiatives. And so throughout the experiment, we're able to quantify their resistance to disease based on 3 different metrics. We have resistance, or the fraction of individuals of that genotype that once exposed to disease developed visible lesions, where we have a variation across the board. We have highly susceptible and susceptible individuals that almost always got diseased. In the middle, intermediate, sometimes they did, sometimes they didn't. And then at the other end, we have resistant genotypes where they seem to never really get disease. This also aligns very nicely with transmission, or the overall proportion or the speed at which they develop disease lesions. We see the same exact pattern. And then finally, progression, or the speed at which that lesion moves across the fragment, matches exactly as it has for all 3 metrics. From there, we can construct a susceptibility hierarchy where the genotypes on the left-hand side of our tree are highly susceptible and susceptible to the disease. And so if you were to pick individuals that were going to be outplanted out onto the reef, you would say, oh, I want to pick ones from the right-hand side of this tree, those that are resistant or intermediate, that can better ward off disease when they're out in a real-world environment. And indeed, we've done that. These corals have been run through propagation. They've been outplanted out onto the reef. And you know what? It turns out that their susceptibility rankings that we gave them in the lab do match fairly well with what happens out on the reefs. So we are currently outplanting disease-resistant genotypes onto the reef to promote the recovery of Florida's coral reef. So this is a lot. I want to thank you all for your time and attention. Feel free to reach out to me at this email on here, as well as see our website at the AOML Coral Program. I'll be happy to take questions at the very end, but thank you all again.
Aloha, good morning, good afternoon, good evening, depending on where you're located. I'm Brian Hauk. I'm the Resource Protection Specialist for Papahanaumokuakea with NOAA's Office of National Marine Sanctuaries. And thank you all for tuning in to learn more about Condor tumulosa and about biosecurity in Papahanaumokuakea. Okay. In addition to my co-authors listed on the title slide, I want to recognize that the information I'm sharing with you all is a culmination of effort from many, many individuals. That's including those that operate the ships, the small boats, the planes, all that to get us to our survey sites. It truly is a group effort. And on top of it taking a village, it also takes almost a week transit by ship to get to the areas I'm speaking about. So the data driving this research came from less than 2 months of actual field work and have led to numerous publications, research projects, and more that I don't have time to elaborate on today. This presentation is going to give you all a crash course overview of biosecurity protocols for Papahanaumokuakea, while highlighting a new species of red alga that we've discovered and going over some management strategies we're implementing to further combat its spread. I'll keep you guys posted with this outline as I go through the talk so you can follow along. Okay, first though, I would like to start with an introduction to place. The area outlined in blue here is Papahanaumokuakea Marine National Monument. The monument is co-managed by 7 co-trustee agencies and has many overlapping boundaries and protections within it, including its recent National Marine Sanctuary designation. Regardless of all those titles, this place is Papanaumokuakea, and it's one of the few UNESCO World Heritage Sites that's recognized for both its natural and cultural resources. The monument's home to over 7,000 species, many of which are endemic, endangered, or threatened. All right, before continuing, I want to revisit this graph that you've seen today a lot. And I want to stress the importance of prevention and highlight some of the biosecurity pre-access policies that we have in place for Papanaumokuakea. When I talk about Chondria later, we're going to be moving up to this containment area of the curve. So as you know, as I told you, accessing Papanaumokuakea is not logistically easy. Isolation is helpful from a biosecurity aspect, but managers must ensure that permanent activities don't inadvertently act as vectors for new species introductions. So in order to do so, we have a series of pre-access permit conditions, inspections, and various BMPs that apply to any permanent vessel that's entering the monument. As you've seen in some of these other slides, presentations, the majority of alien species are transported either by ballast water or biofouling. So because of this, all vessels and ancillary equipment are required to be free of alien species, that's including rodents, not just marine stuff, in order to comply with permit requirements before they're cleared for entry into the monument. As we've learned, whether looking at terrestrial or marine ecosystems, the obvious key to alien species management is prevention. And this is especially important in the marine environment, as it's typically even more difficult to eradicate marine organisms once they're introduced and established to the point of detection. That's why NOAA puts such a large emphasis on prevention by conducting these hull inspections on any vessel before they're cleared for entry into the monument. Okay, so now that you've got some background on Papanaumokuakea, let's explore Chondria tumulosa, where it is and why we should care. For this section, we'll be focusing on the shallow waters around the three most northern atolls, first starting with Monavai or Pearl and Hermes Atoll, then moving up to Koahelani or Midway Atoll, and finally Holaniku or Kure Atoll. We have two species of troublesome algae we are actively managing within Papanaumokuakea, Chondria tumulosa and Acanthophora spicifera. Today I'm going to be focusing on Chondria, but I do want to highlight its partner in crime that's listed below on this slide. All right, as you'll hear, Chondria tumulosa is new to science and its origins are currently unknown. So because of these uncertainties, you'll see words like nuisance to describe Chondria, as they're less confined in their standards with regard to their origin. Another assumption is that atolls with higher biomass correlate to an earlier date of introduction. That too is unknown and could be driven by ecological factors rather than time. So in August of 2019, a major outbreak of an unidentified red algal species was observed during a research cruise to Monavai. This is a unique situation where a new species was first discovered only after it went invasive. After a return and upon further investigation, it was learned that the species was actually first noticed in 2016 on a different cruise that NOAA Fisheries was operating. This is an excerpt from their cruise report that's showing that Chondria was tentatively identified as something else from the genus Laurencia after it had been collected and subsequently stored in a freezer at NOAA's facility. Here's some other photos of some autonomous reef monitoring structures showing the overgrowth of Chondria tumulosa at Monavai from 2016, showing that there was already some substantial matting taking place. So when we got back, the samples from 2019 and the ones that we found in the freezer from 2016 turned out to be the same thing. And they were described as a new species by Sherwood et al. In 2020 and subsequently named Chondria tumulosa. The species was new to science and is cryptogenic in origin, meaning that at the time there was no record of it existing anywhere else in the world. Once again, this is very strange for something that's acting as an invasive species. The next 3 slides are going to summarize the distribution of Chondria within Papua-New-Guinea to date. In 2016, Chondria was first noticed on the north-northeast and southwest areas of the atoll. That's a common theme that you'll see among the 3 atolls here, and it's assumed to be a correlation driven by trade wind direction and the subsequent water movements driven by that. As of 2024, Chondria has been found on all sides of the atoll and within the lagoon. This atoll, Manavai, has the highest biomass of any of the other atolls, and imagery shows that it's been there since at least 2015. Next, Kauai or Midway Atoll. Chondria was first discovered there in 2021 by our marine debris partners on the northeast back reef. Further surveys conducted by NOAA in 2022 and 23 show that it was fairly abundant on the east, north, and west sides of the inner atoll. We've had limited surveys on the forereef zones, but low levels have been found in the northeast, north, and west sides of the atoll. And I just want to highlight here, you'll see the red circle there. That's where Acanthophora specifica was found within the monument. Okay, moving on to the last atoll. In 2023, we found cryptic assemblages of Chondria on all sides of the inner back reef around Holaniku. Growth was very low-lying and seemed as it was kind of growing in equilibrium, or maybe it was indicative of very early stages of introduction. Its presence was again confirmed in 2024, but there were no signs of increased growth. Once again, limited surveys have been completed on the forereef zones, but Chondria has been found on the western sides of those outer reefs. Okay, so now we're going to move on to ecology on these next few slides. So like any invasive species, understanding the reproductive system of Chondria tumulosa is a critical component of developing management strategies for this alga. The results from this study suggest that Chondria tumulosa mostly reproduces by asexual fragmentation, and that it has the potential for spreading to other nearby atolls. You can note in the photos here the spinous secondary branches and the intertwined morphology, which allow it to grow into these thick mats. These mats readily fragment, producing clones with spinous branches that can be transported through the water column, where they can snag on other substrate or organisms. The next few slides here are going to show what appear to be the various stages of overgrowth. This photo shows a typical kind of healthy reef. And what's interesting about Chondria growth is that it's often very patchy. When you're out on spur and grooves surveying the environment, it can dominate one spur, and then you move to an adjacent area and it's absent. This close-up of this Poeciloperid head, you can see this coral head's being overtaken by Chondria. It's hard to deduce if the Chondria caused the mortality or if the coral colony was already dead and the Chondria got snagged and moved in. But it's fairly common to see varying stages of degraded coral health with Chondria overgrowth like this. On reefs with higher infestation levels, you'll start to see these mats interconnecting and the individual coral heads get covered up as it fills in all the pukas or the little spaces in the reef. And this process continues until the entire kind of reef or spur is overtaken. You're left with nothing but a monoculture carpet of Chondria tumulosa. It's not just the benthos and the coral that Chondria grows on. It can also be seen growing epiphytically on native algae. In this slide here, it's a close-up of a native species of Turbinaria ornate with Chondria overgrowing it. And these bundles can be found drifting and could also be another vector for the way that Chondria is spreading around. These native algae were the main locations Chondria was found at Holaniku, indicating how cryptic yet competitive it can be. Here's another example of Chondria growing alongside a native Padina species. You can see the Chondria coming up from underneath and around the perimeter. A closer look here, you can see Chondria's plasticity. and you'll note the changes in its morphology and pigmentation. Okay, a couple more images here showing more of this mat formation. When you remove the mat, you can see all the dead reef beneath it. And then notice when you look at that dead reef, you can see the attached algal fragments, which remain after this mat comes off. So you can imagine, you know, Hawaii gets these huge winter swells in the winter. And you can imagine what happens on the northern sides of these atolls and envision how these mats are ripped off and these tiny fragments get left behind that could seed the next cycle of regrowth. When these mats do break up, they can move down current as these kind of marine tumbleweeds, and they can accumulate into huge amounts of biomass, which I'll show you in these next slides. These tumbleweed accumulations are so large that they can be seen and tracked from space via satellite imagery. If you note, looking up at figure A, you can see how Chondria tumulosa has also been found rafting on the surface, either as a single species or intermixed in a conglomerate with other native algae. We're not sure what causes some of these algal mats to float or sink, but they can move and be transported either way. These tumbleweeds oftentimes accumulate on shore. Here you can see endemic Hawaiian monk seal kind of making a bed out of a pile of chondria. And we just got back from a cruise in last September, and NOAA's technical closed circuit rebreather team also found some of these tumbleweeds as deep as 67 meters or 224 feet. It's unknown if the species can survive in this situation, as the lower depth limit is still unknown for the species. Okay, so now that you know about chondria, let's talk about something else. Where did this stuff come from? How did it get here, if it's not native, that is? So we had a few theories about how Chondria tumulosa may have made its way into Papahanaumokuakea. One being that it rafted there on some type of marine debris, possibly from the 2011 Japanese tsunami, which conveniently matches the potential time arrival of an introduction. This has been proven before elsewhere and would make sense for chondria, but Japan has a very detailed account of what algal species are present there in its waters, and so far, Chondria tumulosa is not known from that area. Beyond tsunami debris, Papahanaumokuakea has a huge marine debris problem, as it lies right within the giant Pacific garbage patch, and it's inundated with an estimated 50 metric tons of debris annually. Some of these nets have been marked with satellite tags and have been shown to have the potential to move between atolls, potentially spreading any hitchhikers that might be attached to them. We have found some of these derelict fishing nets with chondria growing on them. Likely that's been locally sourced given the surrounding habitat, but nonetheless, it shows the potential for transfer among reefs or atolls by marine debris and fishing nets. So digging a little bit deeper here, our partners over at UH use publicly available oceanographic data and software to assess how the potential dispersal of Chondria tumulosa is affected by particle properties and oceanographic conditions. They targeted the capacity of both reproductive tetraspores as well as drifting fragments to disperse. Their model results indicated fragments modeled as rafting on marine flotsam, such as marine debris, settle at a much higher rate than tetraspores. This is helpful to theorize the spread within Papahanaumokuakea, but it doesn't really tell us where chondria came from. So in order to further investigate this, the lead author ran the previous models shown in reverse, with monovi as the starting point. This backtracking experiment was used to better pinpoint potential source populations and can potentially simplify source sampling efforts using technologies such as environmental DNA, which I'll touch on in the final section. Oh yeah, with the question mark here, remember that section over the Marshall Islands. Okay, this brings us to our final chapter, which will cover some management perspectives of this problem. A working group of co-trustee agency reps and subject matter experts have been working since 2019 to better guide science-driven best management practices designed specifically for preventing the further spread of this newly discovered algal through permitted activities. Future trials will feed into the development of new adaptive BMP strategies. These condor-specific BMPs complement pre-existing biosecurity requirements already touched on for entry and work within this special place. Now, finding condor's native habitat and developing early detection rapid response capacity make tools like eDNA very valuable. University of Hawaii's PhD candidate Patrick Nichols, pictured in the top right there, developed eDNA capability for condor, and that has greatly increased our early detection abilities and reduced the survey effort required to locate and find potential source populations. Utilizing these eDNA technologies, UH has now found that Condor tumulosa is also present at Majuro in the Republic of the Marshall Islands. It was growing cryptically there, similar to at Ho'olaniku in Papahanaumokuakea, further muddying the waters on this species' native range. It's not known whether or not this species is native to the Marshall Islands or it was also introduced to the area. Management actions have been developed for other problematic red macroalgae species in the main Hawaiian Islands, but further discussions on the logistical and ethical complications of doing this in Papahanaumokuakea require more thought. Papahanaumokuakea is considered a pristine area that isn't subject to overfishing, eutrophication, or many of the other anthropogenic threats that reefs in the main Hawaiian Islands face. Removal and or biocontrol efforts are a massive logistical undertaking, but managers are also discussing pilot studies and strategies to fill that knowledge gap. Okay, well, hopefully that crash course gave you the who, what, where, and why for Condor tumulosa, as well as some insight into the current management strategies that NOAA and their partners are looking at. And with that, I'll kind of close it out and we'll save our questions for the Q&A chat or the end of these presentations. Thank you very much. And I will pass it over to Steve.
Thank you. Appreciate it, Brian. Thanks for all that. That is one nasty customer you're dealing with out there. And I'm going to be talking about lionfish, which are in a slightly different phase of invasion than some of the other things you've heard about. My name is Steve Gittings. I'm Chief Scientist with the Marine Sanctuary Program, and I'm at the headquarters level, formerly a sanctuary manager, but I've been at headquarters for a good long time now. But I work on lionfish a bit on the side as a research project and trap development and things like that, but also in just general basic dealing with the problem across the sanctuaries that are affected by it. And, the invasion of lionfish from the Indo-Pacific into the Western Atlantic happened about 40 years ago originally. And it's a case of we made our bed, now we have to learn to live with it. And so I'll be talking about three aspects of living with it, which really involve controlling it, populations, commercializing lionfish, and counting on Mother Nature for handling the rest of the problem. That's what I'll talk about. Lionfish are beautiful animals. I've heard them called a cross between a porcupine and a butterfly. But their invasion story is interesting because it has as much to do with people as it does with nature and the invasion itself. Lionfish were introduced, like I said, 40 years ago or so now. It seems like a long time, probably by aquarium owners who saw them eating all the other fish in their aquarium and didn't have the heart to chill the animals to death or humanely get rid of them in some other way. So they released them into nearby canals around their house, probably in southeast Florida. And they remained there for 15 years or so, were detected, but nothing much happened. And then their population exploded around 2000. And they quickly started covering a lot of ground and gobbling up fish everywhere they went. And in some places removing or reducing biomass by some 60% in the early days. That we worried that they would affect food webs, across large ecosystems in the Western Atlantic. They would affect fish health by, consuming fish that would otherwise be cleaning other species at cleaning stations, and even algae cover by eating herbivores of various kinds. So their potential impacts were really widespread. This is an invasion series that you can see the year in the upper part of the slide here and watch it progress over the years. The first 15 years not doing much. There was a lag period, which is pretty common, I think, for invasive species. But in 2000, you can see this, the population exploding to the north and to the east a bit over into the Bahamas. And then by 2005, covering that area. By 2010, covering most of the Caribbean. And by 2015, really expanding through the Gulf of Mexico and across the entire range of suitable habitat. So quite rapid once it started, a period of about 15 years. And you can see their range is probably going to extend well into South America. They were initially prevented by the Orinoco and Amazon river plumes from reaching the southern parts of South America, but they breached that in around 2020 or so, and now are extending into Brazil, even, and probably will fill in all the gaps down along the coast of Brazil. And this is a slightly different version of the invasion curve that you saw before, but pretty similar, showing more customized to the lionfish, the years in which the specific events happen. But it followed the invasion curve to a T, really, as most other invasive species do. And it was a big concern, a huge concern for us, because the tropical Western Atlantic had not seen a species like lionfish before, an invasion from a species like this, a fish like this in the Western Atlantic. And it did have severe impacts over that first 10 to 15 years of the invasion. It was successful because lionfish grow so fast, they mature in less than a year and become reproductive, and they're prolific breeders. Every 3 or 4 days they reproduce, and a single female, as you can see, can put out millions of eggs every year on a continual basis for years at a time. And they're also excellent hunters. Native species are not familiar with lionfish, so they don't fear them, and leaving them highly vulnerable to being preyed on. The lionfish, unfortunately, are also generalists, so they put a lot of species at risk. Over 170 different kinds of species have been identified in their stomachs over the years by different groups. And you can see their impact. Just a few lionfish can really tear up native population. The cost is significant in certain respects, but some of them have not been realized to the extent that we were worried about. We really thought that they would start eating commercial species juveniles and have a delayed impact eventually on those large animals. That hadn't really been shown yet, although there have been some studies showing that they do affect the prey of commercial species. So the debate's still out on that a bit. There was a study recently published I got the paper today. I think it was just came out, published in Barbados about that they had not seen impacts on commercial species. So that was interesting. But I think the jury's still out on that. Certainly their impacts on non-commercial species have been documented in a number of places. But they also have other impacts on commercial species. They reduce catches in traps of species that are targeted by fishermen. And of course, the envenomation, when you get stung by a lionfish, you know it. And some people are quite susceptible to pain and, reduce the capacity to work for several days. So fishermen can be put out of work and lose money that way as well. But, divers quickly realized when the invasion started that lionfish were pretty easy to capture, and they could protect the reefs if they could get rid of lionfish. At least that was the hope. So we became the predominant predator in the early days of the invasion and remained that way up until quite recently. So they also realized that lionfish are good eating, so that they went after the meat as well. And we've been quite a formidable predator for about 20 years now. Jeff McGregor wrote in a Smithsonian article in 2018 that we are a ragtag army that's fighting back against lionfish. And hunting has made a difference. You can see quite clearly in a lot of places with regular culling and derbies and competitions of other kinds where lionfish are being removed at a significant pace, that the impacts on native species have been reduced. So culling does work. But we do also know that lionfish go down to 1,000 feet of depth. So we believe that eradication is not possible. They cannot be yet. effectively removed below scuba depths, really, not in any significant way, but in shallow water they can. But that deep water population is very high in places we've looked. So extermination of this species or eradication of this species in the Atlantic is simply not going to be possible, which means we're going to have to find a way to live with them. So how do you live with this beautiful fish? And how do you stop it from continuing the wave of destruction that it has already exhibited in shallow water environments? Most agree that continued culling is one of the solutions. There's no question about that. And we can keep populations in many places below impact levels on native species. And what about commercialization? We think there's a lot of potential for that. There already has been shown, if we're going to be forced to live with lionfish, we should try to commercialize them if possible. That could mean a number of things. Ali Al-Haj, the guy laying right here, realized early on, he invented Zookeeper and started a company out of that. So product-wise, and he started making money and it did very well with Zookeeper and eventually sold the company. But zookeepers are still out there, and just about every hunter I know has one. So the proportion of people out there buying these things is pretty high. So products is one way. Commercial fishing, another obvious one. In shallow water, people that have good skills and good with hunting, like Rachel Bowman here, and are dedicated to hunting, have been able to build businesses and sell lionfish commercially to seafood distributors and restaurants, mostly at a local scale. There's not a consistent and large enough supply to supply a national market yet. And I doubt there ever will be unless we can figure out how to catch lionfish in deep water more effectively than we can now. But so that even though there might be demand at the national level, there's not supply. But so while there's not a large market, lionfish meat is really excellent food, and chefs do enjoy working with it. But lionfish really are more than food, as you can see with what Ali Al-Haj has done, and also the development of a jewelry market for lionfish. This is a really cool example because Phil Karp, the guy shown in this picture, was able to help underprivileged women in numerous countries in the Caribbean start some of their own businesses by helping teach them how to not just work with jewelry, but allow them with their artistry to develop their own products. And he helped them develop markets. He came from World Bank and helped them learn how to start small businesses, and they've done that. Some of these women were in really desperate circumstances financially and personally, and pulled themselves out, and their stories will just make you cry. They're wonderful people and now doing some really good work on their own, starting their own businesses. Arv Chavda and some partners started a business on lionfish leather, buying lionfish and adding value to landed fish by buying leather buying the skins from hunters and creating leather from lionfish. Now, lionfish are pretty small. They've diversified since they started this company into Burmese python and Asian carp, other invasive species, and are doing really well with high level fashion, high quality and high cost fashion designers. And so they're in quite a different level of market than some of the other small markets for lionfish. But this is a really exciting business and it's doing really well. There's a lady named Lisette Coos who started what she calls a total concept restaurant that organizes and supports fishing, lionfish fishing hunters in her community in Curacao. They sell and teach lionfish art and fashion and jewelry making. And they have exhibits in their store and to do educational programs. Really beautiful work she's doing down there, bringing the community together on lionfish control and teaching people how to help protect Caribbean reefs. So there is some potential for lionfish commercialization, but how much potential there is really depends on the answer to the bigger question of, will nature start to work her magic and figure out a way to help control or to control lionfish on our own. That's a really interesting one. Things like starvation, predation, diseases, parasites, all those are ways that nature typically controls invasive species or any species in nature. They're all potential controls. We haven't seen evidence of starvation among lionfish who may have eaten too much of the environment around them. We haven't seen evidence of that. There was a time period when some diseases started to develop on lionfish and reduced populations locally in certain areas, but there was only one real episode of that. So it doesn't seem to be a persistent control. We tried to teach large groupers to eat lionfish by spearing them and feeding them to groupers. We did notice that they had a difficult time swallowing them sometimes. So that's not a great meal, even though the meat might be good and it might be nutritious for them, but it can't be comfortable. And we also didn't see a lot of natural predation emerging from that training. We did see some other consequences where sharks would start coming in, bothering and harassing the hunters. So it became dangerous. So that's not really a recommended practice anymore. So we haven't really seen that take over and become natural predation. But that might be changing based on some things we are seeing now. A few years ago, and this is a continuation of that curve beyond what you've seen so far. A few years ago, we started seeing a decrease in populations in a lot of places that did not have regular culling. It's much to our surprise. And there seemed to be no explanation for it. And these were lower abundances than were expected, obviously, but they've also persisted, especially in natural habitats. On artificial reefs, it's a quite different story that's for a different talk. But on natural reefs, abundances seem to be down. Predation might be occurring across a number of species that we have seen occasionally to prey on lionfish, but not that much and not consistently. But it might be occurring and divers are just not reporting it. Maybe it's night hunting that we're not seeing and so on, but could actually be that adult predation is occurring in lionfish. And there may be some adaptation that could be taking place, although that's a big question mark. Nobody's seen it yet. But I was in a store last summer and I saw a beautiful eel that I didn't recognize and some lionfish in another tank. And the store owner told me he had to separate the lionfish from this eel because this eel was eating lionfish and regularly. And I asked him, where's this eel from? And he said, well, Indo-West Pacific, which is the native land of lionfish. So that was really exciting because I'd never heard of this happening before. He also said that although we all assume something was eating lionfish in their native range, we always get that question. But this eel was only eating the body of the lionfish and leaving the head behind and leaving the spines behind. So it's figured out a way, and I don't know exactly what that is, didn't see it prey on lionfish, but it's adapted its behavior in native land to be able to eat lionfish. Now, if animals could do that in the Atlantic, that could be a game changer for consumption and control, natural control of lionfish. And there's another possibility that I think is probably happening and we're just not seeing it. And that's large fish and maybe some smaller fish preying on juvenile lionfish, which are much smaller than the adults, much easier to swallow. And they're also pre-adults, so they haven't become reproductive yet. So that would be a really effective way to limit the populations. And it could explain why we're seeing this dramatic decline in lionfish in a lot of places. Consumption, not just of adults, but of juveniles, which really would have a good effect. So whether that's, divers wouldn't be seeing this very well because it's hard to see a juvenile lionfish from more than a few feet away. So you're unlikely to see this happen, but it could easily be happening. And if it is, I think we're really onto something with natural controls. I was doing some surveys in the Cayman Islands this last couple of weeks, and we did juvenile lionfish surveys and adult surveys. And we were seeing quite a lot of juveniles near shore, hidden in seagrass around rubble. And the farther out we swam in the bays, the less we were seeing any lionfish, small or large. So it's very possible that once juveniles move out of the shallow habitats where the predators are not, into areas where there are predators, they may be getting eaten before they even become adults on the reef. So that could be a very good sign that that this kind of predation is happening. So we could be in what we call a post-peak lionfish phase. And while we understand the characteristics that drive the beginning part of this curve going up to the invasion peak, that post-peak period is really not well understood. But a lot of invasive species end up in some kind of boom bust cycles that happen during their post-peak time. And lionfish might be in that area now. We don't know whether they're going to settle in certain places at a level where they're going to be continually destructive to the environment, or somewhere down lower at level where they're maybe moderately disruptive or variably disruptive across different time frames or different locations, or they just they almost dissipate and become a non-consequential animal in the ecosystem and just equilibrate and become part of the architecture, but not a very impactful one. We don't know where they're going to end up, and it could be that they end up in different levels at different places. I think nature will probably eventually be able to control lionfish without our help, even though we probably will still be involved with commercialization aspects and so forth. And may already be doing that to some extent. But how it plays out in different places could reflect the condition of the environments that lionfish are in. And it follows that healthy populations of large fish will increase the potential for lionfish control through, resistance. If there are a lot of healthy smaller fish in the ecosystem, that could help ecosystems recover following the invasion of lionfish. So it could affect both the resistance and the resilience of those ecosystems by having healthy fish in the ecosystem. And a lot of places, as you know, do not have large or small fish in any abundance. And so those places are probably more vulnerable to continued impacts. So I think there's a lot to be said for strong regulations, much stronger than we have now in a lot of places in this invaded region. And I think if we don't do that, they're going to continue to be impacted by lionfish. If we do strengthen regulations, a good chance these places can recover to the extent that they will be able to. And speaking of, invasive species, I'll finish with this. Let me leave you with a thought, and that's to remind you not to put too much blame on lionfish here, because they certainly didn't choose life in an aquarium, which is how they got the problem started. And you've seen this invasion curve already, but this is the still rising human population curve overlaid on the invasion curve. Our population, that yellow dot on the left, is on its way up to about 10 billion people. And it's supposed to peak in about 60 years and probably start to drop in about 80 years. So our impacts have not even peaked yet. The question is, how big will that peak be? And will it drop when our population starts to drop after 80 years? Who knows the question of that? So while the ocean may be able to adjust to lionfish, one of its own, it seems to be having much harder time adapting to the terrestrial invader that we are, that seems hell-bent on controlling nature. And so I'll leave you with that, just to help you, make you think about some of the bigger problems we have beyond lionfish. Okay. Thank you for your time.
Okay. So I think we have our first question here. It was actually to Val, but I think also Michael might be able to share some input here to please explain the stony coral tissue loss disease tracking for the Panama Canal through vessels and next ports of entry, especially with the next port visited being Hilo or American Samoa. And are there other ports at risk? Should all vessels undergo treatment before entering the Pacific?
Thanks for the question. So the way that we are tracking target vessels, which is in particular those carrying ballast water at this point, the ones I referred to with the Coast Guard, is through the National Ballast Water and Ballast Water Clearinghouse, which is implemented by the Smithsonian. And so all folks at this point in time, all of the vessels that are transiting to U.S. ports and are carrying ballast water have to report their ballast and where they are exchanging it or treating it. And so that includes the last 5 ports. And so the Coast Guard has been screening to see when vessels have transited from affected jurisdictions heading to one of the island jurisdictions. At this point in time, we are not able to track simply vessel. We haven't been really looking at just vessel traffic, which could be the whole fouling component. And we don't have any ability to track the international traffic. So something may come from one of those jurisdictions and visit our neighboring islands, and we might not have records of that. So there are a lot of gaps still to be addressed in the regional biosecurity of how things may be moving. As far as whether they should undergo treatment, I think that is kind of a loaded term. There's water exchange, or there's treatment options. And at this point, there's still a need for more evaluation of how effective those treatment options are. And I will leave it open to Michael if he wants to add anything additionally to that.
Sure. Thanks, Val. Yeah, there are a lot of other different treatment approaches that we can test. Some of them are a bit more challenging to implement in a lab-based system. UV is very easy to implement because there are scalable UV systems, but say chemical treatment is another commonly used ballast treatment approach. And it's very difficult to then strip those chemicals out of the water or neutralize them to then expose it to corals. So it has been difficult to actually test that out. But I would say even if those approaches are 100% effective in removing potential pathogens from the water, there's so many additional sources that exist on a ship and inside the surfaces of the inside of the ballast system, the hull. I mean, Val showed a whole infographic with everything. So I think it's perhaps a bit more challenging than that, unfortunately. And to assess or to answer your final question is there a treatment specifically with this disease? No, there is not. And part of that lies in the fact that we don't know what causes the disease. So we don't know what targeted treatment to use.
Well, thank you for that. And then maybe, I know we received a couple other questions about the establishment of healthy coral nurseries to preserve coral populations. And then also a question about upscaling the outplanting of the resistive coral, that you had written some answers. If you'd like to elaborate on those for the group, that would be great.
Yeah, I'm happy to summarize them because they're very similar. So basically the questions were, have we established nurseries of healthy corals, and then can we effectively scale up propagation and outplanting of disease resistance in populations? And the answer is yes to both. So as the disease was moving its way through Florida's coral reef, a massive undertaking across a series of different agencies got together to do a coral rescue effort. It was at a scale that had never been done before. Tens of thousands of coral colonies were rescued from the reef in advance of the disease making it to that area. And for the last 10 years now, they have been held in captivity in land-based systems. So that's not only taking care of them, but that's also starting to propagate them, to spawn them, so that we can build up those numbers that we have. So eventually, when it's safe to put them back out on the reef, we can. And we're doing the exact same thing with resistant genotypes as well, propagating them on land and putting them out on the reef when we can. The biggest limitation for all of that is infrastructure, personnel, and funding. These kind of multi-year propagation pipelines cost a lot of money and take a lot of time because corals grow so slowly. So it's something that the state of Florida, I think, has done a remarkable job dedicating resources and expertise to do it. But as far as Caribbean-wide restoration response, we have a lot more infrastructure that needs to be developed before we get to that point.
Great, thank you. And then I think our last question we had here was for Dr. Sturtivant. And that was regarding whether the climate mismatch that you mentioned with your horizon scanning, does that now hold feasible given the increasing changes to water climate?
We used in ours roughly a 5-year climate, and then we revisit in 5 years. So we have not really done the long-term climate projections in our horizon scanning and risk assessment. They're much more short-term. But yes, I would say probably if we see significant changes in water temperatures, some of those species will need to be revisited and consider whether they're now a threat.
Thank you for that. And then we just received another question for Steve. Have you heard of the ROV used by Atlantic Lionshare? Just saying, we tested this ROV in Bermuda that can reach mesophotic deeper reefs, and it can provide a potential solution for areas that are too deep for tech divers. That's from Taylor Tucker.
Yeah, I am aware of that, and a couple other people have tried that as well. And there have been some interesting ROVs and other robotic type solutions for deep water collections. Some involve electrocution or capture, suction samplers or spearing. One had a laser idea that never came to fruition, but I thought that was an interesting one, laser beams killing lionfish. But there were a lot of them out there, and they've all, I mean, some of them actually do have potential or have had potential. They've demonstrated successfully. Their problem with most of them so far has been volume, being able to get enough animals quickly enough or consistently enough without having a whole fleet of things out there constantly, which would cost tremendous amounts. And trying to make them affordable to things like fishermen who might be able to commercialize the catch if they are incentivized to go get them. So it's a matter of being able to collect enough things. That's limited the implementation of deep water collection devices so far. The electronics is also another thing, as you know, they break down a lot. But so now the Lionshare one, I haven't looked at in detail. I had seen a few that led up to that, but so I haven't looked at that one to see if it's got that sort of volume potential that other ones haven't had. That would sure be nice to find one that does, because so far everything from traps to other robotic devices have not reached the potential we know they'd need to really have an impact on the full population of lionfish. Same with submersibles. There are people that want to put subs down and suction sample constantly lionfish, but there's just, you can't catch enough quick enough.
Well, thank you for that. And then Brian, if you'd like to share too, what do you think? I mean, you did a great job covering, chondria and kind of our concerns about it spreading in those areas. But what about for, other island communities, those of us on the Pacific coast, what should we be on the lookout for with this species?
That's a good question. We still have a lot to learn about, if we knew its native home range, we would have more information about, what environmental factors control where it can be established. So I think just keeping your eyes out for anything new and raising the alarm if you do see it, because the stuff got there some way. I'm pretty sure it can grow in pretty low light levels. We've seen it very happy at 80 feet on the reef and up to subtidal kind of intertidal areas. So just keep your eyes peeled. That's my advice, and raise the flag if you see anything new and interesting.
Thank you so much. We've gone a little bit over time, but this has been a really wonderful panel and a great way to start National Invasive Species Awareness Week. So I want to thank all of our speakers. I will share our call to action slide for a moment here for those of you that do want to contact your representatives and senators to get support for this legislation. And we do hope to see you at future webinars. We have on tomorrow, we have the USGS. We'll be covering some of their research and partnerships. And so we look forward to seeing you there. But thank you to everyone for joining us today. And we will see you later in the week.